Hello. This presentation explains many of the key players in the process of digestion in mammals. Those players include locations, enzymes, and the materials that are moving to accomplish the task of providing animals with those macronutrients we talked about in class, and also the micronutrients. When you think about mammalian digestion, you have to think about the mammalian digestive system. And one of the things you may notice is that in this mammalian digestive system, there's going to be fewer compartments compared to other vertebrates. For example, things like the crop and the gizzard are not seen here. There's basically a mouth, the conduit for food from the mouth to the stomach, which is the esophagus. And then there's going to be intestines for digestion, for absorption, and eventually also for excretion. One of the things we notice in the mammalian digestive system, however, is that there is going to be a greater number of glands that are going to be contributing enzymes and other secretions to help with the process of breaking down food. The first stage in digestion is going to be taking place in the mouth. Usually people don't imagine that the mouth is a place where not only food is broken down mechanically with the action of the teeth, but there's also going to be a little bit of enzymatic digestion. Salivary glands can produce saliva which contains amylase, and salivary amylase, we learned in class, is going to be important for breaking down polysaccharides into smaller polysaccharides and even some disaccharides such as maltose. Maltose is a union of two glucose molecules, so it's another example of a disaccharide. In the saliva, there's also going to be mucus, water, salts, and glycoproteins, and this entire mixture is designed to help food move, glide more easily through the esophagus and down into the stomach. When you're chewing your food, mixing it up with saliva, now we know the components of saliva, you're going to be forming something we call the bolus. This bolus makes food manageable so you can swallow it. The pharynx allows for passage of the food to the esophagus and from there to the stomach. We do have a special gland, which is called the epiglottis. I'm sorry, not a gland. It's actually more like a valve called the epiglottis that is going to prevent food from entering your respiratory tract, the trachea. And the reason this is important is because if people accidentally allow food to go into the respiratory system, the breathing system, it can lead to a form of pneumonia. That in hospitals, they call this food aspiration, and that is not a good thing because of the risks pneumonia can pose on a patient. Eventually, what would happen is that the food is going to arrive at the stomach. The stomach is going to have a mixture of materials with a pH of about 2. And that is going to be largely as a result of hydrochloric acid. I'm going to attempt to enlarge this illustration so you can see a little bit better the cross-section of a portion of the stomach. By now, you may have already started to work on your lab dealing with histology with tissues and cells, and you may recall that a cross-section of the stomach may look like a finger-like projections. And what you see over here is that it's not really fingers, but it's more like pits that are forming the stomach. These gastric pits are going to allow for different cells to provide their contributions to the process of digesting food. For example, you're going to recognize here there are some cells, they're highlighted in yellow, and these are going to be known as parietal cells. There's also going to be chief cells, which is going to be the majority of these cells over here. And then there's also going to be mucus cells, the ones that are highlighted in red. Upon closer exploration, we can discover that the contribution of these cells is going to allow for a mixture that makes it easier for our bodies to break down and digest proteins. For example, parietal cells are going to be in charge of secreting separately hydrogen ions and chloride ions. Once these two materials go into the gastric pit, they will combine to form hydrochloric acid. Hydrochloric acid here in the stomach has two functions. It does help break down some of the food by chemical reactions, but equally important is going to be that hydrochloric acid is going to activate a compound known as pepsinogen. Pepsinogen is going to be the precursor of an active form of an enzyme known as pepsin. So with hydrochloric acid present, pepsinogen is converted to pepsin. Pepsin is described as a protease, and you can see that name right here on my slide. A protease is going to be a protein digestive enzyme. The prote part makes reference to a protein, and ASE, as you know, is going to be the ending we provide for the names of enzymes. And what this pepsin will do is that it's going to cleave, it's going to break protein molecules into smaller polypeptides. Parietal cells that are releasing this hydrochloric acid, well, they actually, remember, release the hydrogen ions and the chloride that eventually become hydrochloric acid, are going to be activated usually by pressuring your stomach. When you eat something, there are going to be special nerve cells that act as pressure receptors, and that is going to be signaling the brain, and that in turn sends a signal from the brain to the stomach, activating the production and the release of hydrogen ions and chloride to allow for this transformation of pepsinogen into pepsin. Mucus is going to be also important in this case. Remember, we have these highlighted in brighter red. Those cells that are producing mucus, the mucus allows for a protective layer, essentially preventing our own digestive juices from damaging the cells that are lining the stomach. There's also going to be in the stomach a coordinated set of contractions. These contractions allow for the food in the stomach to mix with the pepsin, and this is an action that is known as churning. One of the things you will notice also as you are working on your histology lab, in addition to these columnar epithelial cells, among which we will find the parietal cells and the chief cells and the mucus cells, there's also going to be behind them layers of smooth muscle, and smooth muscle is important for any organ in the form of a cavity. Compression of those walls in an organ like a cavity, including intestines, that is going to be the action of smooth muscle. And I thought it was a detail worth mentioning to you, since you may be at the same time you're studying this chapter on animal nutrition, also adding your details to the study of tissues and cells. Digestion in the small intestine is also going to be important. Before I come here, there's going to be another detail that I wanted to mention. Yes, the detail is going to be about the sphincters. And so these sphincters, again, are going to be like valves that will be preventing the contents of the stomach, which by now can be known as chyme. Chyme is going to be this highly acidic material from going back into the esophagus. Sometimes people suffer from damage to this sphincter, which is known as a cardiac sphincter, due to the proximity to the heart. And if this is not working well and food moves from the stomach to the esophagus, they end up suffering the consequences of heartburn and eventually even gastroesophageal disease, also known by doctors as GERD. So the cardiac sphincter is going to prevent food, chyme, from going back into the esophagus. And this one down here is going to be known as a pyloric sphincter. I was hesitating there for a moment. The pyloric sphincter prevents chyme going from the stomach into the small intestine before it's ready to go there. That's a detail that I was forgetting, but I wanted to mention to you too. The small intestine is going to be probably the main place where chemical digestion takes place. And this is going to be an organ that is divided into three sections. There is going to be the duodenum, ileum, jejunum. The duodenum is the first part. This is where food first enters after leaving the stomach and first enters the small intestine. This is where chyme from the stomach will be mixing with other digestive materials coming from the pancreas, from the liver, from the gallbladder, and also from the small intestine lining cells. The pancreas is going to be responsible for producing a wide variety of enzymes, as you know by now. For example, proteases. By now you know protease means a protein digestive enzyme. Proteases like trypsin and chymotrypsin come from the pancreas. They're actually activated in the lumen, meaning the lining of the inside of the intestine of the duodenum. The nature, the pH nature of these secretions coming into the small intestine is going to be basic, and that is a wonderful thing, because remember that chyme coming from the stomach is going to be acidic, a pH approximately 2, and when you mix this acid with the basic alkaline nature of the materials that are being poured into the duodenum of the small intestine, that is going to lead to a neutralization reaction. And as I said, that is a wonderful thing for allowing food for continuing to be absorbed, nutrients to be absorbed, and preventing damage to portions of the intestine further along the digestive process. Bile is usually produced by the liver, and it is going to be stored in the gallbladder. Bile is going to be essential for the digestion and absorption of fats. Epithelial cells lining the small intestine are also responsible for producing different digestive enzymes, and some of those we don't have to be too concerned about. There is a form of amylase, carboxypeptidase are examples of enzymes that are secreted by the lining of the small intestine. The combination of all of these enzymes with the chyme, with the food that moved from the stomach, is going to result in a greater breakdown of macromolecules like lipids and polypeptides and complex sugars into manageable size molecules that can be absorbed by the small intestine. That is going to be the main function of the jejunum and the ileum portions of the small intestine. Digestion is going to be mostly what happens in the duodenum. One essential nutrient that has to be absorbed by animals is going to be fats, and they have to go through a particular process. In the small intestine, you're going to notice that cells often have these projections known as villi. These are going to be extensions of the plasma membrane that are going to be allowing for an increased surface area and the absorption of fatty acids. Notice how that triglycerides, which is a more sophisticated name we have for fat molecules like oils and saturated fats, those triglycerides must be broken into fatty acids. You see two individual fatty acid representations. Here you see a glycerol molecule with one fatty acid. This is a monoglyceride. And that is how they need to be so that they can be absorbed by the membranes of these cells in the small intestine. Once they move into the cells lining the small intestine, these fatty acids are going to rejoin the monoglycerides, forming triglycerides once again. Many of these triglycerides are going to be packaged into units that are going to be surrounded by phospholipids, like the ones that are forming a plasma membrane. There's also going to be cholesterol, and also there's going to be proteins that are going to allow for the formation of these units, chylomicron. Imagine this chylomicron as a pocket containing many of these triglycerides. Chylomicrons then migrate to the opposite end of the cell lining the small intestine, opposite to where the triglycerides, fatty acids, monoglycerides came in. And then they're going to be released into a lacteal. The lacteal is going to be a lymphatic vessel that will allow for chylomicrons containing triglycerides to travel over to the blood, eventually from the blood to the heart, and from the heart to be distributed to all of the parts of the body where these triglycerides may be needed or for storage, as it is in the case of adipose tissues. And here's one more time the view of those enzymes that we want to remember for the exam. Remember that we break them down by digestion that happens in the oral cavity in the mouth, what happens in the stomach, And then in the small intestine, we're going to be concentrating mostly on those pancreatic secretions like amylases, trypsin, chymotrypsin, pancreatic nucleases, and lipases. And that's the end of this presentation. I'll make one more dealing with particle feeding. See you next time.